Hi, this is Dr. Hartejupal. So I am interested in gene transcription regulation. What is a gene? Is gene the coding region of the DNA which codes for amino acids and subsequently proteins? Most of the people think of genes as those which form proteins, but there are genes which also form only RNA, and those RNA never get translated into proteins. So 25,000 protein producing genes and 5,000 at least, and I'm thinking that it should be at least 10,000. There should be more to be found. Since RNAs is so important in regulation of the whole cellular machinery, not only DNA, but entire metabolism. So the protein coding part of the gene are the exons, and they constitute only 1 to 2% of our DNA. And remember, our DNA has 3 billion base pairs. So what is the rest of the DNA up to? Half of our DNA have unique sequences and half have repetitive sequences. The repetitive sequences are of various types. They can be retrovirus-like elements, which are literally ancient relics of retroviruses, or retroviruses originated from it. And then we have the line elements, sign elements, and also these DNA elements which jump as a whole DNA rather than first getting translated into RNA and then through the transcriptase go back to reform the DNA. So the other half is the genes, the gene coding area, the RNA coding area, and the regulatory element. Just look at the exons here and look at the large amount of regulation going on. So essentially, this is the start point of transcription. And look at the number of inputs that are needed and number of and the machinery. This is like part of it is machinery, part of it is input. Here are the inputs coming, pioneer factors. These are the transcription factors here. Various kinds of inputs coming in, and they get translated into gene. The more, let's say you have only one active enhancer, the rate of translation into mRNA is going to be low. And if we have 6 enhancers active, the rate is going to be faster. So this gene is in a loop. Genes are in loops. So the loops are closed by CTCF dimer complex, which is a CCTC sequence binding factor. And then we have a cohesin, and there are genes, or there can be multiple related genes in it. So here we have the cohesin CTCF, and then we have the gene, and the genes are studded with nucleosomes. They are not just DNA. Our DNA is married to proteins called nucleosomes over which it winds around. And then, of course, there are other proteins coating on top of it. So more than half the mass of our nuclear material, the chromatin, which contains DNA, is proteins. So the DNA here has nucleosomes. So what's a nucleosome? Nucleosome is essentially the DNA turns around the nucleosome 1.7 times and 147 base pairs, and then it is an octamer with 8 histone chains, and these are N-terminal chains. This is where the modification happens, and these get modified in various ways during transcription and also during transcription suppression. And this is how the DNA is packed. And this is a chromatin, and it can be really packed tight. In heterochromatin, it can be coated with polymers. So how many types of chromatin are there? There's constitutive heterochromatin, which covers that 50% of the DNA which we talked about, which does nothing other than jump around. That jumping is very important for evolution. Big jumps in our evolution were created by those elements, but they are harmful elements. They can destroy the genes if they kind of jump in between the introns and exons here. So they have to be kept repressed, and they are repressed in tightly wound chromatin covered with polymers. Then we have facultative heterochromatin, which is also similarly condensed. It also has the same condensed structure, yes, but it has less coating, so more accessible. So the pioneer factors can come in. They're the pioneer transcription factors and wedge in here. And the pioneer transcription factors are the OCT, SOX, HOX, NANOG, ALF. They are the stem cell, and they promote stemness of a cell. So those, and then we have poised bivalent chromatin, which is, you can say it's called bivalent chromatin. It is it has even less coating. It is still repressed, but it can be activated by a strong stimuli. And what are we talking about? Which kind of stimuli? For example, when we wound ourselves, the fibrocytes convert into fibroblasts and fibromyocytes or fibromyoblasts. So how does the transformation happen? It's the bivalent chromatin genes. They get turned on again by pioneer factors because the DNA is wound around nucleosomes here, and the nucleosomes usually prevent access by access to the transcription factors. Now, how are you going to access this? The DNA code is hidden here. It's over tightly wound. The DNA is tightly wound around the histone, so it cannot really attach. It's a surface where it's difficult to attach. You want to unwind this, and the way to unwind is the pioneer factors. They come and wedge inside this and loosen it. And then more factors can come and attach and then keep on loosening it. And there's a chain of events which I will talk about, which ultimately leads to depletion of the nucleosomes and the, in fact, in the housekeeping genes, which are the active euchromatin, this entire region is can be free of nucleosomes. The CPG islands, they can be completely free of nucleosomes, the GC box, CC80, even the Tata INR, DP, MT, MTE, DCE sequences can be completely free, and the polymerase along with gender transcription factors will be already attached, ready to go in these genes, and then you get a mild stimulus from transcription factor, and it immediately starts producing genes. So these are the genes involved in our metabolism, daily metabolism. So 60% of our genes are active, 40% are inactive or repressed, and this is in any particular differentiated cell. The combination of active and repressed genes creates the different tissues. In our body, we have a brain tissue which looks nowhere close to what muscle or a bone looks like. However, it's the same genes in every cell of the body, and it's just their varying combination. Like some are the 40% which are repressed, that those are different in every tissue. And the ones which are active, obviously the housekeeping genes are active in all the tissues, in most of the tissues, but then there are certain set of genes which are active in only particular tissue. So this is in nutshell I've given you a synopsis of what a gene is, and my next video will be how a gene is controlled by each of these molecules. It will be extensive, 1 hour talk. Thank you.